Hey guys, today we're going to talk about the membrane structure and how structure allows permeability. So this is actually my second take because I did a bad job the first time. So my cards do have writing all over them. Don't let that overwhelm you. We're going to use structure to explain selective permeability. Selective permeability is the quality of a membrane. Selective permeability means that only some things can come in and out of the cell. The level of permeability determines what comes in and what comes out. What can pass through our membranes and how do we figure that out? We're going to build a model of the cell membrane. It's going to be called the fluid mosaic model. If we break this apart, we can figure out exactly what that means. Fluid, things are in motion. Things are constantly moving in our membrane. It's like a mosaic because it's made of a bunch of different parts. It has a bunch of different moving parts, the fluid mosaic model. The two main stars of this model are going to be the phospholipids and the proteins. We already know from our biochem chapter what these words mean. It's going to be really important that we remember that phospholipids are amphipathic, so they are both polar and nonpolar. This is a really important piece of our explanation of how our membrane provides selective permeability. We're going to connect the word amphipathic to an explanation of how small and uncharged particles move through the cell membrane using simple diffusion. We're going to use this part of our model to explain simple diffusion through the membrane. The second co-star of our model is going to be proteins embedded in the membrane. Some of them go all the way through, some of them do not. They allow a type of diffusion that's called facilitated diffusion. It can also be called transport. Now, who gets transported? Particles that are either too large to pass through simple diffusion or charged, even if you're small and charged. You still need the help of proteins to get through the membrane. If you need the help of proteins to diffuse through the membrane, then that type of diffusion is called facilitated diffusion. So we have two different types of diffusion through the membrane, both of which happen through selective permeability. There's 3 guest stars in the membrane today. We're going to talk about cholesterol, glycoproteins, and glycolipids. These sound like 3 scary words, but you already know them, because glycoproteins are exactly what they sound like. They're half protein and half carbohydrate. That's where the glyco comes from. It sounds kind of like glucose, which we know is a carb. Now, same deal with tiny word number 3. It's part lipid and again part carbohydrate because it's a glycolipid. We're going to take some itty-bitty side notes about what are these guys, what do they do for our membrane. We're going to take a brief peek at the cell wall because not all cells have the cell wall, but some of them do. Who has it? What does it look like? And what does it mean for permeability? First, let's take a look. This picture of the cell membrane should look really complicated because the membrane is complicated. It's made of many, many moving parts, which is why this is called the fluid mosaic model. Part number 1 that we're going to take a look at is a phospholipid. This is its head. This is a tail. Let's zoom in on one. This phospholipid has been zoomed in on. Let's just go through in a little tiny review. What are they made of? The hydrophilic head is polar. It needs to be polar to be hydrophilic. What is it made of that makes it polar? It has a phosphate group and a glycerol. That makes up the entire hydrophilic head. The hydrophobic tails are made of fatty acids that are either saturated or unsaturated and connected to the glycerol through ester bonds. Most important takeaway is that any phospholipid has to be amphipathic. It has to be polar and not. And this quality is what causes our membrane to form the phospholipid bilayer, because these hydrophobic tails turn around and hide from water, and so they face each other instead. This environment is entirely hydrophobic because it's made completely of these fatty acid tails that are also hydrophobic. So the inside of any cell membrane is completely hydrophobic and water-fearing. But alas, we have the hydrophilic head. So what does it do to the quality of our membrane? It makes both surfaces, the outside and the inside surface, are both charged. They both have a charge because they're made entirely of these polar phospholipid heads. Let's talk about the function. Pretend for a second that our membrane is made completely only of these phospholipids. It doesn't have any of these proteins embedded inside that we haven't even talked about yet. These are going to allow only for simple diffusion. Some things can pass through the bilayer on their own without the help of proteins. That's why this diffusion is so simple. These particles need to be small to fit through tiny gaps in the membrane, and they also need to be non-polar so that they can survive in this environment, because this environment is, again, hydrophobic. That's going to be simple diffusion. You don't need any energy, and you can just pass right through the membrane. Now, things like water are a little too big and a little too polar to just pass right through the membrane, but they still need to get in and out of our cells. So how are they going to get through the membrane? They need the help of proteins. Some of the proteins look like this one. It's a channel. It's always open, and this particular channel allows water through. Here's our water molecule. It's called an aquaporin. It has such a specific name because it has such a specific function. It only allows water through. That's why membranes aren't just permeable, they're selectively permeable. Depending on what proteins exist in your membrane, that could change what could pass through your membrane at any given time. Any diffusion that needs you to have a transport like this one, this guy's ugly, this transport protein. If you need help, if you're polar or you're too big and you need the help of this protein to protect you from the hydrophobic environment, then the process of you passing through the membrane is facilitated, and it's facilitated by proteins. There's still no energy involved. Now there's some weird stuff hanging out in this membrane. Let's talk about it. Here are glycolipids and glycoproteins. These are embedded in the membrane, but don't think of them as fixed, because remember, this membrane is fluid. So this is like a snapshot. If we had a video, everything would be floating all around in this, constantly moving. So things that would be moving include not just the phospholipids, but everything embedded in the membrane, like glycolipids and glycoproteins. You can see that both of these kind of stick out of the membrane. They're used in a few different processes. One example is going to be in our immune system. Some glycolipids and glycoproteins are used to recognize different surfaces, different bacteria, different other cells. They can attach to different things, and they can do that because part exists inside the membrane and the other part exists outside the membrane. So what's the difference between the two? The glycoprotein has a protein embedded in our membrane, a protein that may or may not pass all the way through. But the glycolipid is almost like a phospholipid in disguise because it lives among them. It's a part of the membrane on both the inside and the outside because it has these two fatty acid tails that live inside the membrane. Now there's something else living inside this membrane. Who are these fatty acid imposters on the inside of our membrane? This is cholesterol. Why would we want our membrane to have cholesterol? I thought cholesterol was a bad thing. Well, it turns out that it's not. There are two different things that could happen to your constantly moving fluid mosaic membrane that would be really bad. One of them is getting too hot. When things in motion get hot, their speed of motion increases. So that means that phospholipids are moving around faster and faster, and so are any of these embedded proteins. And that could be bad if gaps in between these structures grow and grow as the particles move faster and faster. That could change the permeability of our membrane. So to keep this from happening, the cholesterol is inside the membrane to prevent that kind of breakage when things get too hot. It holds the membrane together even when things are moving at really high speeds. Now, it's kind of like Goldilocks. We know how it helps when things are too hot. How does cholesterol help when things are too cold? Now, even though the membrane is fluid and in constant motion, when mobile things and particles get too cold, they start moving at lower and lower speeds. That could cause our membranes to become way too tightly packed for things like simple diffusion through the membrane to occur. That would be really harmful to us. So that's why we want cholesterol in our cell membranes. It keeps these phospholipids just a little bit far away from each other in certain places. That prevents them from becoming too densely packed. So again, it's like Goldilocks. Cholesterol keeps damage to our permeability from occurring when things are either too hot or too cold. Now, we promised that we would not only talk about how the different components of our membrane cause selective permeability, but how does our cell wall affect permeability and why? There are 3 different types of cell that could have a cell wall. The first we're really familiar with. This is the plant cell, and their cell walls are made of cellulose. Remember, that's why it's really hard to eat a tree, because we can't digest the cellulose in their cell walls. In bacteria that might look something like a fuzzy potato with a tail. This is a really bad drawing of E. coli. A bacteria like E. coli has a cell wall too, but this one's made of peptidoglycan. Fungi also have cell walls. These are made of chitin. And although these are made of 3 different things, they have 2 very similar functions. One of them is to give structure to the cell, to keep it really strong, to keep plant cells square, to keep bacteria cells shaped like potatoes. It also places additional constraints on permeability. Because remember, even if we do have a cell wall, on the inside of that, we also have a membrane. Our membrane is selectively permeable, but our cell walls are also selectively permeable. So anything that needs to enter the cell needs to be able to pass through both the wall and the membrane. So cell walls provide additional constraints on permeability. Just as a quick review, we talked about 2 different kinds of diffusion in this video. Neither one of them required energy because things spread out. They move from an area of low concentration. They move from an area of high concentration to low concentration without using any energy. This happens all on its own. Diffusion from high to low concentration. There's 2 different types across our cell membrane. There's simple diffusion, which can only happen if you are small and nonpolar. It's simple because the only 2 things you need are something to diffuse across and the phospholipid bilayer. It's as simple as that, but some things need a more complicated form of diffusion. It's called facilitated. They need some help because they are polar or large. They can't fit through the membrane usually, and even if they could fit through gaps between phospholipids, They couldn't exist in this hydrophobic environment. That's why they need the protection of things like protein channels, just like aquaporins. Proteins help polar things get through the membrane, and that's why their diffusion is called facilitated. There's still no energy. Tune into our next video where we'll talk so much about transport that does require energy.